Today, we're talking about a promising new approach to cancer therapy using an anti-podocalyxin CASMab.
Podocalyxin? What kind of protein is that?
It was originally identified on podocytes, specialized epithelial cells in the kidney. Podocyte means foot cells because they have long, tentacle-like extensions. Podocytes are essential for blood filtration, and podocalyxin helps them form and maintain their characteristic structure. Podocalyxin is a cell surface glycoprotein. In other words, it is a protein on the cell membrane that carries a large amount of sugar chains.
So it's one of those heavily decorated proteins on the outside of the cell.
Exactly. Those sugar chains give podocalyxin a strong negative charge. It helps keep cell surfaces from sticking to each other too easily. In that sense, podocalyxin helps maintain the shape and surface structure of certain cells.
So it's one of those super important kidney proteins, then?
Exactly. However, podocalyxin is also highly expressed in a variety of cancers, including pancreatic cancer.
Whoa, pancreatic cancer too?
Yes, and that matters, because increased podocalyxin expression has been associated with greater malignancy and poorer prognosis.
So if a cancer has more podocalyxin, there's a good chance it's nastier.
That is the concern. Podocalyxin interacts with the actin cytoskeleton and appears to promote cancer cell migration and invasion, which may contribute to aggressive tumor behavior.
So all we need is an antibody against podocalyxin.
Actually, antibodies against podocalyxin have already been developed because podocalyxin has been used as a diagnostic marker for kidney disease and for cancer. In fact, our lab developed anti-podocalyxin antibodies and showed that administering them could suppress the growth of oral cancer cells.
So the antibody already existed. And it really does have anti-cancer effects, so let's use it for antibody therapy.
No, we can't.
Why not?
Because of the possibility of side effects.
Does this antibody also bind to normal cells?
Every antibody has some side effects, but what matters is the location and severity. Podocalyxin is also present on the endothelial cells of blood vessels and lymphatic vessels.
That's bad news. Blood vessels are a problem. If we use the antibody, the blood vessels would be in serious trouble before the treatment even worked on the cancer.
That's right. For that reason, podocalyxin had been considered useful as a diagnostic marker, but hardly anyone viewed it as a realistic target for antibody therapy.
Aw, come on! It has anti-cancer effects too.
But there is a way to solve that problem, isn't there?
Of course, Brilliant Me has figured it out. If we make a CASMab against podocalyxin, the blood vessels should be safe.
Our lab has previously developed several CASMabs targeting podocalyxin.
Whoa!
As we can see here, the podocalyxin CASMab could bind to these cells derived from pancreatic cancer. In comparison, binding to lymphatic endothelium-derived cells is very limited.
Great!
Also, CASMab could inhibit the growth of pancreatic cancer cells.
It seems so promising.
Yes, and today's study takes that idea one step further.
Ooh, even further? Now I'm paying attention.
Instead of using the antibody directly, the CASMab was also used to create CAR-T cells.
So now we're turning CASMab into a CAR-T weapon. Okay, hit me with the basics again. What exactly are CAR-T cells?
CAR-T cells are T cells that have been genetically engineered to express a chimeric antigen receptor, or CAR. This receptor combines the antigen recognition part of an antibody with intracellular signaling domains that activate T cells.
So you take a T cell and basically give it a brand new target sensor.
That's a good way to put it. Once infused into a patient, those engineered T cells can recognize and attack cells carrying the target antigen.
So instead of just waiting around, they go straight for the target.
Exactly. And compared with giving an antibody by itself, CAR-T cells can keep working for a much longer time.
Right, because a regular antibody wears off, but CAR-T cells can stick around and keep fighting.
Yes, a conventional antibody has to be administered repeatedly because its effect is temporary. CAR-T cells, by contrast, can persist and continue exerting anti-tumor activity.
That sounds seriously powerful. But then, why doesn't the immune system just wipe out cancer on its own?
Our immune system does have ways to attack cancer. Cytotoxic T cells, also known as killer T cells, are responsible for destroying abnormal cells. However, cancer cells often find ways to escape.
Ugh! Of course they do. How?
Some exploit immune checkpoints, which act like brakes on T cells. Others reduce or hide the surface signals that help T cells recognize them.
That's so underhanded. Cancer cells really do fight dirty.
And there is another, more fundamental issue. The immune system is designed to eliminate what it perceives as non-self.
If cancer cells do not appear sufficiently foreign, they may not be attacked efficiently.
Right. One important factor is the presence of neoantigens. Cancer cells often accumulate mutations because their DNA repair systems are impaired. Those mutations can produce proteins with altered amino acid sequences.
Since those mutant proteins were not originally present in the body, they can be recognized as non-self.
Right. Such mutation-derived cancer-specific proteins are called neoantigens.
Hold on! Neoantigens are cancer-specific too, right?
Yes, they are.
Then what's the difference between a neoantigen and the epitope recognized by CASMab? They both sound cancer-specific to me.
Neoantigens arise from random mutations, so the exact mutated protein often differs from one patient to another.
Ah, I see. So one patient's neoantigen might not even exist in another patient.
Exactly. That makes neoantigens highly individualized targets, and therapies based on them often need to be tailored to each patient. In contrast, the epitope recognized by CASMab is not necessarily based on a patient-specific mutation. Instead, it reflects a cancer-specific state or structure of a protein that can be shared across many tumors.
Oh, that's a big difference. So CASMab could work across cancers that share that same cancer-specific form.
That is the advantage. CASMab-based targeting may be less patient-specific and potentially applicable across multiple patients, as long as their tumors share the same cancer-specific epitope.
So neoantigens are more personalized, but CASMab could be a lot more broadly usable. Nice! But even if cancer cells make lots of podocalyxin, regular T cells still wouldn't automatically attack them, right?
Correct. If the protein sequence is essentially the same as in normal cells, the immune system may not judge it to be non-self.
Aha! So that's why CAR-T cells are such a big deal here.
Yes. If you build CAR-T cells using a podocalyxin-specific CASMab, you can create T cells that actively attack podocalyxin-expressing cancer cells.
And because the targeting part comes from CASMab, those T cells should still leave normal cells alone. Beautiful!
That is exactly what the researchers wanted to test. The team tested if those cells could attack podocalyxin-expressing cancer cells safely and effectively. This study was a collaboration with Professor Shin Kaneko's lab at the Center for iPS Cell Research and Application, Kyoto University.
All right, let's see the result.
First, let's look at the effect of the CAR-T cells on cells expressing podocalyxin. The target cells were cultured together with the CAR-T cells, and the researchers measured how much cell death occurred. They tested several different ratios between the two cell types.
And?
These were the results. Please note that on the x-axis, the left side represents a higher proportion of CAR-T cells. The red and blue lines are the CAR-T cells. The blue line represents CAR-T cells made using CASMab, while the red line represents CAR-T cells made using a conventional, non-CASMab antibody. The green line represents T cells without a chimeric antigen receptor.
Both types of CAR-T cells showed cytotoxic effects.
But in glioblastoma cells in which podocalyxin had been knocked out, no cytotoxicity was observed.
So the CAR-T cells weren't just going wild. They were really targeting podocalyxin.
Exactly.
Okay, but now comes the scary part. Were normal cells safe?
That was the critical question. The researchers then tested cytotoxicity against normal vascular endothelial cells. CAR-T cells made from a conventional anti-podocalyxin antibody caused damage to those normal endothelial cells, but the CASMab-derived CAR-T cells showed little to no such cytotoxicity.
Amazing! So the regular version hurt normal vessel cells, but the CASMab version basically didn't. The biggest safety problem might actually be avoidable.
That is the significance of the study. And they checked tumor suppression too. They compared CAR-T cells made from the original mouse CASMab with CAR-T cells made from the humanized CASMab. Both showed anti-tumor effects compared with the control treatment.
Very nice.
There was also another interesting finding. The CAR-T cells based on the humanized CASMab appeared to suppress tumor growth more strongly.
So humanization may do more than just improve safety as a drug format?
Possibly. In this study, tumor volume began to increase again after about 60 days in the mouse CASMab group, whereas growth remained more strongly suppressed in the humanized CASMab group.
So the humanized version may have had better sustained efficacy.
Here's a result from another previous study examining the effect of CAR-T cells targeting podoplanin. In that case as well, CAR-T cells based on a humanized antibody showed stronger suppressive effects. Incidentally, it has been found that this mouse antibody actually has higher affinity for the antigen than the humanized one.
Then it's not just a simple matter of stronger binding. So maybe humanization has some additional advantage in CAR-T design.
It may, but the number of examples is still limited. So more research is needed before drawing a firm conclusion.
Still, that's a really interesting clue.
It is.
So let me see if I've got the big picture.
Go ahead.
Podocalyxin is overexpressed in many cancers and is linked to aggressive disease, so it looks like a promising target.
Correct.
But ordinary anti-podocalyxin antibodies are risky because podocalyxin is also present on normal endothelial cells.
Right.
CASMab solves that problem by recognizing the cancer-specific form of podocalyxin while sparing normal cells.
Exactly.
And now this study shows that if you use that CASMab to build CAR-T cells, you can get anti-tumor activity without the same level of endothelial cell damage.
That is the central finding.
So this is another major step toward making podocalyxin-targeted cancer therapy real.
Yes. Previous work opened the door to antibody therapy targeting podocalyxin, including possible application to pancreatic cancer. Pancreatic cancer is one of the hardest cancers to treat, and at present, surgery is the only treatment with real curative potential.
So if we could treat pancreatic cancer by targeting podocalyxin, that would be a huge breakthrough.
This study extends that concept to CAR-T therapy and provides encouraging evidence for both efficacy and safety. Of course, further studies will still be needed, especially to confirm safety from multiple angles before clinical application.
But it definitely feels like a promising new step forward.
A difficult target once thought too dangerous may now be becoming therapeutically accessible, thanks to cancer-specific antibody engineering.
Very cool! CASMab-powered CAR-T cells might turn once hard-to-target cancer markers into real therapeutic opportunities.